The aim of our study was to predict cancer development. For this purpose, we analyzed several microRNAs in the serum of breast cancer patients versus 92 healthy individuals. By bioinformatic studies, we were able to assign a microRNA signature designated predictor based upon the expression levels of 5 microRNAs. Then, we validated our microRNA signatures in a cohort of 20 breast cancer patients. Further, we tested our signatures in 60 volunteers women, assigning 11 out of 60 in the cancer group. Moreover, we found several microRNAs associated with prognosis. On the other hand, we analyzed by proteomics the expression levels of differentially overexpressed or downregulated proteins in the serum of breast cancer patients versus healthy individuals. We found that microRNA 16 were correlated with high expression level of the protein CD44, a stem cell marker. Overall, we describe a predictor able to distinguish breast cancer patients versus healthy individuals. The clinical applicability of our predictor should be tested in large women cohort. For this purpose, we plan in the future to increase of serious patients and analyze the microRNAs differentially expressed in the serum levels. Finally, I would like to thank to all the investigators that have collaborated in this study, especially to several physicians from the CAP Alcarcas and Gervasi, from the Vall Hospital, the breast cancer unit, and also from the BIR.